Have you ever looked at something and said, I wonder? This is how scientists look at the natural world. They are curious and they use all of their senses to make observations about nature. Observations are the first step in the scientific method. We all make observations every day, but scientists don't stop simply when they make an observation, but rather they combine that with additional information in a process called inductive reasoning to form a hypothesis. A hypothesis is a tentative explanation for that observed natural event. A hypothesis, however, is only valid if it can be tested. Scientists then use deductive reasoning in determining how to test a hypothesis. Deductive reasoning involves if-then statements. For example, a scientist might reason, if organisms are composed of cells, then microscopic examination of any part of an organism should reveal cells. Further observation sometimes is all that's needed to test the hypothesis. However, other times scientists must perform experiments. Good experimental design is critical. A scientist must ensure that they are testing what they want to test. Many experiments will include an experimental group as well as a control group. The control group is the one that is not being tested. In the experimental group, a single factor or variable is manipulated. It is critical that all factors other than that single experimental variable are held constant. At the conclusion of the experiment, the results or data may be analyzed and the hypothesis is either supported or rejected. It is important to note that a hypothesis is never proven to be absolutely true. New knowledge or sometimes advanced experimental techniques can always prove a hypothesis to be false. For example, the Earth was considered to be flat for a long, long time. Over time, a hypothesis that is consistently supported may be assembled with others to form natural laws, and ultimately into scientific theories. Many investigators do controlled studies in which experimental group or groups receive some treatment. Again, it is important that all factors other than the experimental factor be held constant. The factor that is manipulated is called the independence variable. The result of that manipulation is called the dependent variable. The control group does not receive the experimental treatment and becomes the basis for comparison at the end of the experiment. The following example is a controlled study. This study is based on the fact that nitrogen is a limiting nutrient in many plants. For example, in the spring of the year, many of us apply fertilizer to our lawn. That response, that nice, lush, beautiful green lawn, is a response to the nitrogen in the fertilizer. Also, even though the atmosphere is 70% nitrogen gas, plants cannot use nitrogen gas. Some plants, called legumes, contain bacteria in root nodules. These bacteria are able to take nitrogen gas from the atmosphere, and they convert that into a form that the plant can use. Therefore, these plants do not require additional nitrogen fertilizer. Peas are a legume. Wheat is not. The hypothesis here is that a crop rotation between pigeon peas and wheat will result in residual nitrogen in the soil so that the wheat will not need to be fertilized. The biomass of wheat growth will be measured following growth of the peas to compare the biomass of wheat grown without crop rotation or fertilization. This diagram illustrates the experimental design. The control pot that's shown on the far left of the diagram received no fertilizer. Then there were 3 experimental pots. One of these experimental pots received a very low level of nitrogen fertilizer. Another test pot received a higher level of nitrogen fertilizer. And the third test pot was involved in a rotation between the pigeon peas and wheat. Many times the results of an experiment will be shown graphically. This diagram shows the results of our pigeon pea wheat crop rotation. During year 1, the pot with the lowest concentration of fertilizer really showed no greater biomass than the control pot. This might indicate that applying only a very low fertilizer concentration really nets no increase in growth. The higher concentration of fertilizer did in fact result in increased biomass, while the pot representing the crop rotation did no better than the control pot. If we look at year 2, there was actually a significant decrease in the biomass from the high fertilizer concentration, but we do begin to see significant increases in the biomass of the pot representing the crop rotation. Now if we look at year 3, what happened to the control pot? That would be the little short blue bar. Well, after 3 consecutive growing seasons, there were no nutrients left in the soil, and practically no wheat growth occurred. There were also decreases in the pots that did receive nitrogen fertilizer. What about that last bar, the brown bar? This represents the biomass in that pot where we have now had 3 years of crop rotation. After 3 years of crop rotation, yes, there is significant increase in biomass. So does this support the original hypothesis? That original hypothesis stated that a crop rotation between wheat and a legume, such as pigeon pea, would result in increased biomass of the wheat. So yes, after 3 years, the hypothesis is supported. First of all, a valid hypothesis must be testable. Also, remember that a single variable can be manipulated during the experiment, and that all other factors must be held constant. Also, remember that a hypothesis is never proven absolutely true. And also, experiments must be repeatable. This means that another scientist should be able to arrive at the same result if they follow your exact experimental procedure.